Yeah, thanks so much, and thanks everyone for organizing this meeting and for coming out. I really do appreciate it, and judging by the large attendance here and on the phone, we recognize this issue as being taken very seriously by FDA, so thank you for all of your work looking into this. I first became interested in Mefloquine when I was deployed to Afghanistan in 2007. Prior to that, I was working as an epidemiologist in a surveillance activity for the U.S. Army, looking at electronic records of pharmaceuticals and outcomes and so on. I was only marginally familiar with the concerns related to Mefloquine. I had assumed it was safe, as I'd been taught in the Army. When I was deployed to Afghanistan, though, I quickly realized something was wrong. As we were preparing to board the planes, rifles and backpacks in hand, I was told to reach into a garbage bag and pick out a box of Mefloquine. And the one I picked out didn't have my name on it, and I was assured that it was all the same and everything was fine. And that was the start of my interest in this. The more I looked into it, the more I realized the military was not following its own rules, and certainly not following the rules that it had said it would follow, product insert and so on. And I think if you're familiar with my work, you can see that I've been committed to documenting these deviations from practice. Well, let's get into my presentation quickly. I want to keep lots of time for Bill and the others to speak. Mefloquine is a neurotoxin. I think we should be clear on that by now. And in addition to being a neurotoxin, you can think of it as being a psychotropic drug. And like any neurotoxicant, most of its effects will be subclinical. They won't necessarily meet the definition of a diagnosable illness. There'll be abnormalities, but they'll be vague. They may be poorly defined. Think of it as lead. Not everyone exposed to lead will be easily diagnosed with something clinically, but that doesn't mean that lead is not a potent neurotoxicant. And what makes Mefloquine particularly challenging is that its neurotoxic effects may mimic a lot of other conditions, including things like somatoform and conversion disorders. Up until very recently, I think there's been a lot of doubt that these effects could be long-term or permanent. But I hope we recognize, with the science out there, that permanent neurotoxicity and permanent effects are, at the very least, biologically plausible. And we can talk more about that. And just like lead, neurotoxicity disproportionately affects folks in the inner cities and so on, Mefloquine neurotoxicity disproportionately affects an already vulnerable class of users. Peace Corps members and military and veterans. And they have their own share of problems already, to which now the effects of Mefloquine neurotoxicity are added. These short-term, acute, subacute side effects that we all recognize are associated with Mefloquine, it's best to think of these not as side effects, but as symptoms of a single neurotoxic or intoxicating syndrome. There's structure to these side effects. They're consistent across users. And ever since it was shown in the early 2000s that Mefloquine is a potent blocker of interneuronal gap junction communication, the neuroscience and neurobiology community has been using Mefloquine to mess up neuronal biology in experiments. And from these experiments, I think we can safely conclude that the most likely mechanism of intoxication is blockade of these connexin gap junctions. These are critically important for inhibitory control within the limbic system and brainstem, and some experiments have shown direct effects on downstream dopaminergic tone. So I think if we want to postulate what is the biological mechanism of short-term or subacute effects, it's this gap junction blockade. And a lot of insights into this disease syndrome that it causes can be found looking at the limbic encephalitides. The symptoms of Mefloquine intoxication are very similar to what you see in the paraneoplastic limbic encephalitides. But to some degree, it also shares a lot of symptoms in common with limbic seizures, right? Limbic seizure disorder, temporal lobe seizure disorders. And if we administer PCP or ketamine to some users, you see the same kind of symptoms in some people. And so with these as disease models or pharmacological models, I think we can begin to appreciate the plausibility of Mefloquine as a truly psychoactive substance. So what is Mefloquine Intoxication Syndrome? What is it? Well, we know what it is from looking at the product insert, right? The product insert says, if you experience symptoms of anxiety, restlessness, depression, or confusion, these may be considered prodromal to a more serious event. These are the early symptoms of this developing limbic encephalopathy. And I suspect with rising dose or with more significant progression, you develop more and more higher level symptoms. It begins in the brainstem, moves up into the limbic system, and ultimately moves to the prefrontal cortex. And when it gets up to that level, we see this association with acts of violence and suicide, not unlike what you see with PCP and ketamine. But what makes this particularly challenging, and I think which has led to this being missed for so long, is that this is idiosyncratic. You can administer methadone to everyone in this room, and not everyone will have the same reaction. Individuals that don't suffer this susceptibility will wonder what's wrong with this person that is suffering this susceptibility. It's idiosyncratic. And it's very different than what we see with other drugs, because I suspect it's pharmacokinetic. Now, Dr. Dow, who worked at Rare, incredibly, he was able to publish this stuff in an environment that was very hostile to this type of research. He demonstrated that at high doses, and for the moment, just keep in mind that high dose is high dose in CNS, and CNS concentration will vary across people. So he forced this high CNS dose by massively dosing these mice. But you see this very characteristic neuronal degeneration in the brainstem. And what's going on here isn't entirely clear. It's sort of like what you see with quinolytic acid toxicity. And quinolytic acid toxicity, as we know, is sort of this common pathway by which a lot of diseases seem to cause neuropsychiatric effects. Even cerebral malaria, we think now, is linked to overproduction of quinolytic acid. But it could be other things. It could be glutamate excitotoxicity and some new theories out there to do with gap junction toxicity, deal with blockade of glial gap junctions. But here's what's important. This is not the first time we've seen this focal, very specific type of neurotoxicity with quinolines. This was known as early as the 1940s when U.S. military researchers were investigating the 8-aminoquinolines. And if you're familiar with the history of these drugs, we got most of these drugs from the Germans during World War II, and we were eagerly investigating these compounds as replacements for quinacrine and so on that were used as antimalarials during World War II. And a certain class of 8-aminoquinolines produced these very striking, very scary lesions throughout the brains of affected animals and even humans. And it was associated with this acute intoxication syndrome, prodromal symptoms of restlessness, anxiety, psychosis. So there was a beginning of a syndrome being seen here. If you go back into literature, up until recently when all this stuff was electronically archived, I think it would be very difficult to find this literature. You know, I'll concede that in the 70s and 80s, no one was going to go dust off these journals and read these articles, but now it's all electronic and you can look this stuff up. Read what's highlighted here. It's just remarkable. The entire nervous system was probably affected, but the brunt of the damage was in definite nuclei of the brainstem, and the symptoms and signs reflected this localization very clearly. Schmidt, who was working for the Army, did a lot of work on the neurotoxicity of immunoquinolines. And what's striking is how cautious he was. He was warning us to look carefully for signs of this toxicity, because what he said was, and notice here, he's saying the principal nuclei of the proprioceptive, visual reflex, and vestibulo-ocular cerebellar pathways were affected. What he was saying was, these drugs cause so many symptoms that might mask evidence of this low-grade neuronal injury. So if you're going to be developing these drugs, you have to do very, very, very careful histopathological testing to rule out this type of damage. So Schmidt worked on these drugs for a few years in the 50s, and then I don't know what he did in the late 50s and early 60s, but he was hired again by Walter Reed to look into the next generation drug discovery effort. And instead of the 8-aminoquinolines that were looked at in the 40s and the World War II period, military focused on the 4-quinoline methanols, or 4-methanol quinolines. And for some reason, they didn't put the same effort into searching for this brainstem neurotoxicity that they put into the 8-aminoquinolines. And maybe they should have, right? Because they had early warnings that this same syndrome of brainstem toxicity mixed with sort of a limbic encephalopathy was occurring. Schmidt here says that these doses of this one 4-quinoline methanol, which is not mefloquine, evoke lightheadedness, difficulties in focusing, which should have been a clear indication that something was going on with the brainstem visual nuclei, headache and nausea. So when the first human studies of mefloquine were done, there was more evidence of this brainstem syndrome. All 4 subjects reported transient dizziness and nausea within the first 48 hours. It's been assumed, I think, and not unreasonably, that the nausea was sort of GI-based. There was some local trigger reflex that was causing the nausea. But in light of everything that we know about the neurotoxicity of these compounds, and the fact that the nausea takes some time to occur, it can take 12 hours or more to occur, this is more consistent with a central emetic effect. So, when mefloquine was being developed, what I find fascinating is that the same authors that emphasized the need for this very careful brainstem histopathological testing were very silent about this potential with the methanol quinolines. And they didn't do any, as far as I can see. And even though they recognized vertigo and nausea, lightheadedness, dizziness as evidence of this very dangerous brainstem toxicity syndrome, they didn't follow up on this in the development of mefloquine. And what I find very disappointing is that the folks that should have known better, the folks at Rare that had all this institutional knowledge, the Army, Roche, that knew about this propensity with the earlier drugs, they seem not to have informed FDA of this fact. And as I said, it'd be very difficult to find these journal articles back in the 70s and 80s. You know, this is covered in dust. And how many libraries have these papers? So I found an old transcript of an advisory committee meeting. This wasn't the original licensing. This was in 91 when we doubled the dosing frequency on the basis of those Peace Corps studies, which we can talk about some other day. But it's obviously confusion as to whether the drug's even penetrating the brain. And some of the participants, well, it should penetrate the brain, but does it matter? Well, maybe not. And Dr. Maxwell, who ultimately signed off on the NDA, said, well, we couldn't find anything, and we don't know. But that's actually, that's incorrect. I mean, there was good evidence here, tested on owl monkeys. And I think actually Schmidt was doing these tests. I think Schmidt was in charge of doing the owl monkey tests. It's obvious that the drug was accumulating in brain, and the brain was a major reservoir for the drug. So unfortunately, a lot of missed signals. Okay, so look, so what can we do? Well, we can't undo the past, right? But we can, I think we can clarify a lot of things with the package insert. Unlike a lot of folks that are involved in mefloquine, I don't think mefloquine should be banned completely. It's becoming potentially very useful in treatment of cancer, PML. You know, it could be a very useful chemotherapeutic agent. But continuing, practices of the past is I don't think an option. And let's talk about some problems. Look, the package insert says, ever since 1989, Roche has insulated itself from liability by saying if you develop prodromal symptoms of restlessness, confusion, anxiety, depression, you must discontinue the drug. Must, not should, you must discontinue the drug because these may be considered prodromal to a more serious event, the more serious event being encephalopathy. Well, if you're administering this drug to an infant, how are you going to ascertain that the infant is suffering these symptoms? There is no reliable way to ascertain these psychiatric symptoms in an infant or a child. And I don't know what the cut point is, but a pre-verbal child is clearly not able to accurately express the fact they're not feeling well. So I question whether we can use this drug safely in infants and children. And then I think we need to clarify the mental health contraindications. This has been the source of much trouble over the years because what's happened is that it's been assumed by many users and many practitioners that these psychiatric adverse effects, which are real, biologically, substantive events, these are just latent mental illness that's showing itself in the presence of the drug, or this is just a coincidental manifestation of pre-existing mental illness. The reason I think, epidemiologically, we see slightly more reactions in people with mental health histories is simply because if you're experiencing prodromal symptoms from mefloquine and you already have a history of depression or you already have a history of anxiety, these symptoms are nothing new to you. You're used to experiencing them, and so you're less likely to attribute them to your use of the drug. And what you may do is you may take more of your Xanax, you may take a Valium, like I've seen many soldiers do. You may take some Adderall, like I've seen some soldiers do, assuming that it's not the drug. And that's only going to exacerbate the subsequent toxicity, isn't it? Because you're going to keep on taking mefloquine, it's going to accumulate in your brain beyond the neurotoxic threshold, and then you'll suffer long-term effects. So I think we need to clarify why these mental health conditions are contraindications. And then the other thing is it's sort of like if you go out to a bar and you drink, right? You know you shouldn't drive home if you've had more than a couple of beers. But by the time you've had a couple of beers, your decision-making is affected. Your executive functioning is altered. And you think you can drive home safely. And this is why any good party host will take your keys away before you get to the party, because you can't be trusted to make these decisions once you start suffering the intoxicating effects of alcohol. And it's the same thing with mefloquine. We see this time and time again. If the early prodromal symptoms of intoxication are paranoia and anxiety, and you're in the military, and your commander told you have to take this drug, or I'm going to punish you, right? You're not going to be able to make a very difficult decision to discontinue the drug in the face of this paranoia. And I think, Patrick, later maybe you can speak to how organizational structures and so on affect the safe use of this medication. So just giving someone a wallet card that says, if you experience these problems, stop taking it, I think that's it's a little naive and it's a little simplistic. And then, lastly, we need to explain what this more serious event is. This is a very inappropriate euphemism to include in a product insert. This has also been the cause of much trouble. What is a more serious event? What does that mean? Does that mean a hurricane? Does that mean you're going to break a leg? Does that mean nuclear war? What does a more serious event mean? Well, we know what it means, right? Potentially violent acts, suicidal behavior, parasuicidal behavior, psychosis, memory loss, death, right? So why don't we clarify what the more serious events are in the product insert? If you do that, and you make it clear that this drug is psychoactive, neurotoxic, and some people it penetrates the blood-brain barrier, may accumulate and cause all sorts of trouble in the brain, physicians and their patients will be able to make informed decisions about the use of the drug without having to take it off the market. People will quickly realize, my goodness, this is maybe not for me, But some people will maybe continue feeling they can take it, and that's fine. So, some final thoughts. I'm pursuing some other research, looking into how a group of scientists were exploring the quinolines specifically for their psychotropic effects. There were a group of U.S.-funded scientists that were looking at quinoline drugs specifically for their psychotropic effects, not as antimalarials. And that was occurring in the 1950s and 1960s. We should think of mefloquine as a psychotropic drug with incidental antimalarial activity. And why is that? If you're going to suffer adverse effects from the drug, they're going to occur within 1, 2, 3, or 4 doses, typically. Some people experience them months or even years later, but typically you're going to experience them early on. During that period of time, the drug does not have effective antimalarial properties, right? We know this. I mean, a 250 milligram tablet will produce serum levels about 250 nanograms per milliliter. That's 1/4 of what it really needs to be to provide effective suppression. So you need to take, on average, 3 to 4 tablets for it to be an effective schizontocide. And because of the market serum heterogeneity, some people take 7 to 10 weeks to build up suppressive levels. So it takes 2 months to be an effective antimalarial, and yet that whole time you're experiencing psychotropic effects, vivid dreams at the very least. So think of mefloquine as a psychotropic drug with incidental antimalarial activity. And look, do the math, right? Only about 1 to 2% of this drug, because it's so lipid soluble, it's ultimately found where we need it to be, in the serum, right, or in the erythrocyte. There's twice as much from animal experiments concentrating in the brain, and probably in individuals who are susceptible, much more than that, concentrating in the brain. And lastly, this is not unique to mefloquine, right? Everything I've talked about with mefloquine, we can see in the other quinoline compounds to a lesser or greater extent. And the differences in drugs across the class, I think, are manifested in more or less susceptibility to this idiosyncratic susceptibility. So there's a class effect that's common to all the quinolines, and it's just particularly noticeable with mefloquine. So that's my presentation. I'm happy to take questions.